Hi everyone, good day. This is Dr. Von. It's been a while and I'm very excited again to teach with you the new topics in histopathology. But before that, I just want to apologize with my previous video because when I tried to scan it, every time na nagsascan ako, ang dami ko palang piyok moments. So hopefully for our topics for today, sana hindi ako pumiyok. Mag-piyok counts tayo ngayon. So for our new topics sa ating histopathology, we will be discussing about your automated and rapid tissue processing. And then our next topic will be your principles of staining. So our main references for these two topics, of course, we have your Gregorius pa rin tayo, which is our main reference, histopathologic techniques. And I also get some information galing sa basic and advanced laboratory techniques in histopathology and cytology. If you do not have any copy of this book or ebook, then I can share it with you guys. So for our learning objectives for today, at the end of this video lecture, the students will be able to explain the parts and functions of an automated tissue processor. Then we will try to differentiate the different types of your automated tissue processors, then explain the advantages and disadvantages of it. Then we will also discuss your rapid processing techniques. And for our second topic, our main learning objective is to explain the different staining principles. So ito talaga yung gusto kong ma-learn nyo, is the principle or the concept, because you will have a separate topic naman sa mga different staining techniques sa atin sa histopathology. Then we will try to discuss different procedure of your staining. So let's talk about with our first topic, your automated tissue processing or your automated tissue processor. So Doc, ano po ba yung automated tissue processor? Remember that when you are doing your conventional tissue processing or tinatawag nating manual tissue processing, anong ginagawa nyo? Simply, you have your tissue sample and then you put it to the different reagents. Tapos after a specific techniques or process, of course, nililipat-lipat nyo siya. But with an automated tissue processor, all you have to do is that put that in the machine and ang machine na ang maglilipat-lipat sa ating tissue sample. It makes our job easier and is more convenient for those who have so many tissue samples. So napapadali ang ating work. So for your automatic tissue processing, ano po ang principle nito, Doc? The basic principle is just to transfer the tissue in the different fluid for a specified time in a desired environment. Remember that your tissue processing can be performed manually. So yung tinatawag natin na hand processing. But when there is a large volume of tissue, so in a government institution, I had my first rotation as a medical technologist sa SPMC in a government institution. And my first rotation is really in histopathology. And you will really see a large volume of tissues. Napakadami ng samples na sinasubmit nila. And I remember that they have this kind of your automatic tissue na processor. So since there is a large volume of tissues that need to be processed, again, it is more convenient and much more efficient to use an automated tissue processing machine or yung tinatawag natin na tissue processor. Next, there are two types of your tissue processor. So ito yung first type, yung tissue transfer processor. So from the name itself, tissue transfer. So ano ang tina-transfer? Of course, the tissue. In this system, the bucket of tissue is transferred from one carousel to other after a specified time. There are several containers with your reagents. So ito yung tissue transfer na processor. This one, so let's check for the parts of our tissue processor. Ito yung sinasabi natin na display setting or display button. Dito natin nilalagay yung specific setup para sa ating tissue processor. Tapos itong mga containers na ito, dito natin nilalagay yung ating mga reagents. Tapos within that, meron pa yung basket. Makikita natin, I have a video later on as titingnan natin. Meron yung basket na nakalagay sa loob. Tapos iikot-ikot lang yan, parang carousel. So this is a tissue transfer processor kasi ang tina-transfer is your tissue. Tingnan natin sa video natin. So this is your tissue processor na Leica TP1020. So again, this is the overview of your machine. So dito yung mga display button natin para ma-set up yung ating specific na processing time. And then, this will be the different reagents or container para sa ating reagents, I mean. Tapos, ito yung individual na mga cover ng ating reagents. And then, this is the main cover of our tissue processor. Tapos, siniset up niya yung specific timing for the different steps or process. Diyan yan ini-input. Tapos, ito yung mga lalagyan ng ating mga reagents. Tapos, it will automatically transfer our tissue cassette. So, ito yung basket na sinasabi ko. Yung basket na yan, maglalaman niya na around 30 to 100 cassettes or tissue sample. The basket containing the tissue is submerged to a specific container for a particular time and then transferred to the next container automatically. So there is also a gentle agitation created by a vertical oscillation or by a rotary movement. So nakikita nyo parang umiikot-ikot din siya because we want that agitation. Later on, we will see what is the importance of your agitation. The time schedule and transfer of tissue in each of these containers are determined actually by your microprocessors. Diba? It's a lot easier kasi mas lalong bumibilis ang ating tissue processing. So, now we will have another type of your tissue processor. We have your fluid tissue processor. Sa fluid tissue processor naman, this is a completely closed processor. Here, the tissue is kept in the container and the container is periodically filled with particular fluid. So, in short, this is now a fluid transfer processor. Tingnan natin, nakalagay dito yung mga different na reagents. So, may mga containers dito for our reagent. Tapos, ilalagay natin yung tissue sample natin parang sa isang container or box lamang. Doon natin ilalagay ang ating tissue dito. Tapos, it's a different kasi nga fluid transfer. Anong tina-transfer natin? Ang fluid. So, your tissue sample dito will become static. So, nakalagay lang siya dyan. Tapos, yung fluid lamang yung nag-change. So, every time, depending on what step ka na sa tissue processing, pinapalit-palitan niya lang yung fluid. Yung tissue sample natin remains static. Unlike sa tissue transfer, yung tissue talaga ang lumilipat-lipat. Dito, ang fluid ang pinapalitan. Yung tissue sample natin is just static. So what is the advantages of your fluid transfer processor? Vacuum pressure makes the system faster and efficient. Later on, we will see what is the importance of this vacuum. And then, it is a closed system and there is no chance for tissue drying. Unlike sa ating tissue transfer processor kanina, of course, since open siya, na-open yung takip niya, then there would be a chance for tissue drying. So this is our video. Tingnan natin. Sana naman mabilis na to. Hina talaga ng PLDT, just ko Lord. So this is a solution video, Tissue Tech VIP6 also. Actually, this is a Sakura na machine. I never see anything before ng ganito, pero ang nakikita ko lang talaga is, ang common lang talaga is yung carousel type. Kasi this is a bit expensive. So Sakura Tissue Tech VIP. So again, ito yung ating machine. Ayan. Diyan yung ating mga tissue sample na nakalagay. Yan yung tissue sample, nilagay na lang dyan. Tapos, na-set up niya na sa ating display button or setting. Tapos, ang fluid lang mismo ang pupunta mismo sa ating tissue sample. Tingnan nyo, pinapalitan ng fluid or nilalagyan ng fluid dito. So, after a certain period, the fluid is pumped out from the container containing the tissue. Then, it is again filled with the fluid required for the next step. In this processor, each step can be customized for vacuum, temperature, and time duration. So now, this is now your fluid tissue processor. Very good. So, the time schedule for overnight processing. So remember, if the processor is to run overnight, it should be programmed to hold on to the first ethanol bath and not finished until the next morning so that specimens do not sit in the hot paraffin longer than the time indicated. If the specimens are fresh, they may incubate in formalin in the first stage on the machine. So remember, it is important to not keep the tissues in hot paraffin too long or else they may become hard and brittle. So if we do a overnight processing, dapat nating nakaprogram siya dito sa ating first ethanol bath and not finish until the next morning. So dapat magsa-start muna tayo dito sa ating first alcohol bath to be followed by your clearing and lastly your embedding. Remember, dapat kasi we do not want to keep our tissue in your hot paraffin too long or else it may become hard and brittle. So dapat magsisimula talaga tayo sa alcohol if we do an overnight processing. Kasi if nagsimula tayo sa clearing, what will happen? After paraffin, of course, masastock na siya dito sa paraffin. And then, what will happen sa ating tissue nga? Magiging hard and brittle, and we do not want that. So this is a schematic diagram showing the time schedule for overnight processing. We will start with the first bath of your alcohol during dehydration, followed by clearing, and lastly for your embedding. So now let's talk about the factors that impact the duration of your tissue processing and extent of infiltration. So this is not only true about your automatic tissue processing, but this is also true with your conventional or manual tissue processing. So this is actually in general the different factors. First, let's talk about your tissue density and thickness. So your spongy tissues like your lungs are usually more rapidly infiltrated than your hard and your dense tissues. And also for your thickness, the thickness of the tissue also influences the rate of your reagent diffusion, hence processing time. So pag makapal ang ating tissue sample, it should be optimized for particular processing schedules or alternatively processing times and should be adjusted to accommodate thick, thin, or large tissue blocks. So ang ibig sabihin lang nito, pag makapal yung tissue natin for our tissue processing, of course, we have to adjust the amount of time because it will require more time for the reagent to diffuse in our tissue sample. Kasi pag masyadong makapal, anong mangyayari? Masyado ding mataas ang tissue processing. We have to adjust the tissue processing time. And again, for those with low density, such as mga spongy tissues, such as lung, remember that it will cause easy infiltration of your tissue sample. So yan lang yung dapat nating malaman. Next, agitation. Again, remember sa ating tissue processor kanina, we have a rotary movement that will create a agitation. So doc, ano bang importance ng agitation? So your agitation using your manual or automated processors increases the flow of your fresh fluids in and around the tissues. Without agitation, tissues tend to settle to the bottom of your processing device or become too tightly packed, therefore reducing now your surface area available for fluid exchange. Therefore, anong nangyayari pag walang agitation? There will be poor infiltration ng ating tissue sample. So there will be poor facilitation ng ating hindi mahihirapang magdi-diffuse yung ating reagents para sa ating tissue processing. So we want this agitation to create an increased surface area. Once there is an increased surface area para sa ating tissue sample, mag-infiltrate or there will be increased diffusion of our reagent. Next, temperature. Temperature in the range of 37 to 45. Just remember this ha. Yung temperature na gusto natin is around 37 to 45 degrees Celsius. For a limited time, this can speed up fluid penetration. So your temperature can also enhance your fluid penetration and tissue processing protocol. However, we have to take note that pag lumagpas tayo ng 45 degrees Celsius, nagiging high temperature, and it can cause now your tissue to shrink and to become hard and brittle. Of course, we do not want that. However, if it low or below 37 degrees Celsius, anong nangyayari? There will be low temperature. Pag mababa ang temperature, it increases now the viscosity of your reagents and use in the tissue processing, thereby reducing the rate of your diffusion and increasing your processing time. So we have to maintain a specific temperature so that it will increase the penetration of your reagent sa ating tissue processing. And lastly, we have your vacuum and pressure. Remember your vacuum na diniscuss natin sa ating fluid transfer processor. Diba? It's vacuumized. Remember, a reduced in a vacuumized na ating mga machine, it can cause improve actually the tissue infiltration and as well as your processing quality. It can also aid in the removal of air from a porous tissues. Using your vacuum during tissue processing protocols can reduce the infiltration time when dealing with a dense and fatty tissue sample. How about your pressure, doc? Sa pressure naman, it can also a reduced pressure can increase the infiltration rate and decrease the time and need to complete the steps in your tissue processing. So, we want a reduced pressure and we want a vacuumized na mga machine. So again, let's review ano nga itong mga factors na to. Pag tissue density, pag mga tissue density or mga sample na mga highly spongy such as your lungs, of course, we want a less, it will cause now easier tissue penetration. So in short, i-adjust natin yung time of penetration. Pag sa thickness naman, of course, pag makakapal na mga tissue sample, we don't want that because it will give us more time dapat ang ating iaalat for the fluid to penetrate. For agitation, it will hasten your infiltration. For temperature, again, we want a temperature of 37 to 45 degrees Celsius so that it will enhance also your tissue penetration. Of course, your vacuum will also enhance the quality of your infiltration ng ating reagent. And lastly, we want a reduced pressure so that it will also enhance the infiltration rate and decreases the time for your tissue processing. So these are the factors that you want to remember when you're doing your tissue processing. So what are the technical considerations that we have to remember when doing your automatic tissue processing? So first, your basket and your metal cassette should be clean and wax-free because we do not want to contaminate our tissue sample. Tissues should not be packed too tightly in baskets so as to impede your fluid exchange. Your processors must be free of spilled fluids and accumulated wax accumulation because, again, we don't want any contamination. Your fluid levels must be higher than the specimen containers. Of course, kailangan yung fluid levels or yung reagent natin dapat higher than the specimen containers because we wanted to have a complete penetration ng ating specimen. Kasi pag mas mataas yung specimen container, of course, it will not fully penetrate sa ating tissue sample. Timing and delay mechanism must be correctly set and checked against the appropriate processing schedule. Again, ito naman, a processor log should be kept in which the number of your specimen processing reagent changes, temperature checks of the wax bath, and completion of your routine maintenance schedule are recorded as part of your quality assurance program. So we just want to have a proper documentation. Your gentle washing and minimal thickness of cell layers will prevent the cells from detaching during your staining and make sure that there are enough sections to make a diagnosis and to ensure that the reagents have been applied evenly to your slide. So this is more of the staining. So later on, we will discuss naman sa staining. Of course, again, this one also talks about your staining. Quality of staining can be compromised by inadequate fixation and similarly by poor tissue processing. A good technician must evaluate and determine the processing of choice for each purposes. So, meron tayong special stains or paraffin, frozen, or cell smear preparation. Make sure the glass slides are clean and free from debris, and in general, needle biopsies and bloody specimen should be incubated conservatively, whereas your fatty specimen can be processed for longer than average. So, these are basic na mga technical consideration. So now let's proceed with your rapid tissue processing. So Doc, ano po bang kinaiba sa automated tissue processor sa rapid tissue processor? Kasi nga sa automated tissue processor, yes, it can hasten your tissue processing, but in rapid tissue processor, it will add up or increases or hasten more your tissue processing. Mas bumibilis pag pinagamitan natin ng rapid tissue processor. Kasi sa microwave tissue processor, it reduces the time of processing. The microwave oven usually meron tayong system to control the temperature, meron tayong system to control the time duration of your particular temperature, and a proper exhaust to remove the toxic gas. The microwave processing may be used for all steps of your processing. So, remember, as opposed to your conventional tissue processor, which uses, di ba, meron tayong graded series of alcohol. Pag gumagamit tayo ng automated tissue processing, of course, the step is still the same. Meron tayong graded series from 50%, 75%, absolute alcohol, 95%. Di ba, meron pa rin tayong graded series of alcohol. Meron tayong clearing agent and embedding medium na ginagamit, which can be used in an overnight process. But with the use of a microwave tissue processor, a microwave-assisted processing allows dehydration to be done in only one step and employs a 4-step process involving a single change in your ethanol and isopropanol and followed by 2 changes in paraffin. In short, your microwave tissue processor will shorten the process. Once na-shorten niya ang process, mas lalong binibilisan niya yung tissue processing. Kasi nga, ganito, baka magulat kayo. Kasi nga, Doc, automated nga siya, Doc, diba? So in short, mas mabilis. Of course, binibilisan niya yung steps. Kasi nga, diba, remember, sa conventional or manual step, anong ginagamit natin? Pag manual lamang, yung tinatransfer natin manually. Pag automated, same pa rin. Tinatransfer, pero ang tinatransfer yung machine. Pero the number of steps is the same. Pag gumamit tayo ng rapid tissue processor, specifically your microwave tissue processor, nililesen na yung steps. It does not necessarily be na dapat 3 palitan ng ating dehydration, 3 palitan ng ating graded alcohol, but here, it can lessen your steps. That's why it is na rapid tissue processor. Di ba ang galing? Hindi pa ako nakakita ng microwave tissue processor. This is now your example of your microwave tissue processor. Actually, this is a microwave oven type or kitchenware type kasi para lang siyang microwave oven. So, ito, for example, depending on what step, dyan mo lang nilalagay yung tissue sample sa loob. Tapos, for specific time, meron lang tayong mas nililesen niya yung tissue processing natin. So, your microwave tissue processor, anong principle, Doc? Of course, it uses your microwave. Your microwave, a form of electromagnetic wave-induced heat, when applied in histotechnology, yields a histologic material of similar or superior quality to that provided by conventional processing methods. Itong conventional processing method, this is now your automated or your manual na processing method. So, gumagamit tayo ng microwave. Remember, your microwave processing can be 60 to 80% faster than conventional processing time with an hourly throughput of up to 120 specimens. So you can process up to 120 specimens. So there is a rapid turnaround of your tissue biopsies, provides an excellent morphology, sharp nuclear detail, and your crisp cellular images. So your microwave processing requires very precise maintenance of your temperature. So ito na yung somehow a disadvantage when using your microwave tissue processor. Kasi kailangan mo yun ng very precise maintenance of your temperature. Kasi anong gusto natin sa microwave? It's about your temperature. So we need to have a very precise maintenance of your temperature in which is not possible with your kitchen type na mga microwave oven. So this is a kitchen type na microwave oven whose temperature and output power are not precisely specified, leading to possible overheating and miscalculation of your percentage output power. So, ito lang yung talagang disadvantage niya kasi kailangan natin ng very precise na maintenance temperature, which is very difficult to do naman, di ba, right? So, in your microwave tissue processor, you can also do a staining method. So, hindi lang siya up to your embedding processes, but also your staining method. Take much shorter time in your microwave oven compared to a conventional tissue processing. So, your microwave slides have better contrast, more intense in staining, and less non-specific staining compared with your conventional metal staining methods. However, the optimal temperature for staining for this is around 75 to 95 degrees Celsius. For metallic stains, and around 55 degrees Celsius to 60 degrees Celsius for non-metallic stain. Again, pag metallic stain ang ginagamit natin for microwave tissue processor, kailangan natin ng 75 to 95 degrees Celsius. For non-metallic stains, kailangan natin ng 55 to 60 degrees Celsius. So these are the precautionary measures when we are using your microwave tissue processor. So sections must be thin enough so fixative and dehydrating solutions can penetrate completely. This may increase the time required in grossing of your specimen. So again, remember we have already talked about this one, about your thickness. Dapat manipis lang so that it can easily be penetrated by your reagent. Certain tissues such as brain and your large tissue blocks require additional steps before placing it into a rapid tissue processor. Continuous flow processing eliminate patching of your specimen and necessitate ongoing attention to your instrument. As samples complete the processing cycle, they must be removed from the instrument to accommodate the next basket of your cassette. Your paraffin must also be added to your microwave in a liquid form as microwave energy will not melt your paraffin pellets. Remember, you have different types of paraffin. Meron tayong liquid form. However, pag microwave ang gagamitin tayo or magagamit tayo ng microwave tissue processor, dapat it should be in liquid form because the pellets will not melt in your microwave. And lastly, tissue must be brought to the boiling point of the intermedium around 82 degrees for ethanol and 78 degrees for your isopropanol in the last step to evaporate and the remaining ethanol or propanol. So let's continue with our second discussion, the principles of staining. So sa principle of staining here, I just want you to learn the different concept that you have to understand because later on, marami naman tayong different topics about your staining. Meron tayong staining in carbohydrates, staining of your CNS, mga immunohistochemical staining na may separate topics. So sa ating discussion today, this is just about a concept for you to understand why we use such staining. So more of the principle lang talaga. So again, what is your staining? Staining is your process whereby tissue components are made visible in microscopic sections by direct interaction with the dye or your staining solution. Remember, there is a colored compound used to produce a contrast between your different tissues and cellular components based on their varying affinities for most dyes and stains. Then, morphological changes are more easily identified as well as the different characteristics and structural relationships of tissues and their cells can be evaluated and the presence or absence of disease can be established. Meaning dito na yung lumalabas yung one of the four aspects of disease process, yung morphological changes. Your staining will help us to identify those morphological changes. So what is the purpose? To enhance the contrast and visualization of the cell or certain cellular components under the microscope, to differentiate between your live and dead cells in a specimen, to demonstrate the relationship between internal and external structures of the cells, and lastly, to identify different types of cells. Meaning your staining will help us or aid in the proper diagnosis of our patient. So for example, this picture is an example of your hematoxylin and eosin staining. So let's identify this tissue. Again, this is columnar epithelium. Tapos may presence ng microvilli. So this is probably ano nga to? So let's identify ano to? This is a gastrointestinal, most probably seen in your gastrointestinal tract. And this is seen in your colon. So this is an example of your hematoxylin and eosin na staining. So let's identify the basic concept. So certain parts of the cell and tissues that are acidic in character, your nucleus, so remember, your nucleus is acidic in character, will have a greater affinity for basic na dyes. While your basic constituents such as your cytoplasm take more of your acidic stain. So ganito lang yan guys. Remember your hematoxylin and eosin, your hematoxylin is a basic na dye. Your eosin is an acidic na dye. So again, anong concept na dapat nating ma-learn? Since your nucleus is acidic in nature, it will attract to your basic na dye. Your cytoplasm is acidic, your basic in nature. That is why it will attract your acidic na stain, which is your eosin. That's why your nucleus will stain blue because your hematoxylin is a basic dye, while your cytoplasm will stain to pink to or light red because your eosin is acidic in nature. So the opposite will attract. So again, your nucleus is acidic, will attract to your basic dye. Your cytoplasm is basic in nature, it will attract to your acidic na dye. So let's talk about the different types of staining. So meron tayong tinatawag na histological staining. Dok, ano po itong histological staining? It is a process whereby the tissue constituent and general relationship between cell and tissue are demonstrated in sections by direct interaction with a dye or staining solution, producing now a coloration of your active tissue component. So example nito is your microanatomic stains, bacterial stains, and your specific tissue stains. So pag histological, you are talking about your tissue sample or mga tissue constituent. Pag histochemical staining naman, from the name itself, histo, still you are talking about your tissue sample. Chemical, you are talking about a chemical reaction because this is a process whereby your various constituent of tissues are studied through chemical reaction. So there must be a chemical reaction to occur that will permit a microscopic localization of a specific tissue substance. So for example, meron tayong tinatawag na enzyme histochemistry. So sa enzyme histochemistry, yung active reagent natin, so yung stain na ginagamit natin, will serve as actually the substrate in which your enzyme will react, and the final coloration produced is from the substrate rather than the tissue itself. So there must be a chemical reaction. Another example, this diagram. So this is an example of your periodic acid shift na stain. So later on, your PAS na stain or your periodic acid shift na stain is an example of your carbohydrate staining. So I will have a separate discussion again with your stains of your carbohydrates. So for this discussion, your PAS stain is a histochemical reaction na stain because the periodic acid, your periodic stain na ginagamit natin will oxidize the carbon-to-carbon bind for forming now your aldehydes, which react to your fusion acid, which will form now your magenta color or your purple color. That's why it will specifically identify the substance na ating gustong makita. For this example is your glycogen. That's why your glycogen will have a color na magenta. So mayroon tayong specific na constituent na gustong makita. Because remember, your histochemical, various constituent of tissues are being studied. So ito yung sinastudy natin, yung glycogen for this example. We have also your immunohistochemical staining. So you have a separate topic also for your IHC staining. I guess this will be discussed by Dr. Charlie. Makikinig nga ako dito kasi I never had the chance or opportunity to see IHC staining. Personally, wala talaga ako never pa ako naka-encounter or nakakita under the microscope. So IHC staining is a combination of your immunologic and histochemical techniques. Again, histo kasi we're talking about your tissue, chemical because we are talking about your chemical reaction, and immunologic because we are talking about your antibodies. So we are talking now with your immunologic component. Again, using a wide range of polyclonal or monoclonal fluorescent label or enzyme labeled antibodies to detect and demonstrate your tissue antigen and phenotypic markers under the microscope. So ito yung nakikita natin under the microscope because of the fluorescent labeled or enzyme labeled na antibodies. Diba? It looks so nice. So, I want you to learn the basic principle of your IHC. So, ganito yan. So, Doc, paano po ba namin nakikita ang ganitong kulay? Bakit po lumalabas ang ganitong kulay sa ating microscope? So, may tatlong ways para makakita tayo ng ganitong kulay. Could be your fluorescence-based detection, colorimetric detection, and conjugated antibodies. But this one is an example of your fluorescence-based detection. So this is the principle. Again, you have your protein here or your tissue sample. Ito yung tissue sample. Remember that your tissue sample is composed of your antigens. May mga antigen yan. Since it is an antigen, what will happen? You have your primary antibody. Your primary antibody will attach to that antigen. When your primary antibody is already attached to that antigen, your secondary antibody will attach to your primary antibody. Again, pag naka-attach na si primary antibody sa ating antigen, a-attach na yung secondary antibody. And your secondary antibody contains now your fluorophore. Ito yung tinatawag namin na fluorophore, such as your fluorescein or your rhodamine. Yung fluorescein or rhodamine, ito na yung nakikita natin na kulay sa microscope. So because your secondary antibody contains now your fluorophore or fluorescein, a reaction will occur. Another reaction that will happen is your colorimetric detection. Sa colorimetric detection is still the same. Meron tayong antigen or your tissue sample natin, tapos primary antibody will still attach on it, and your secondary antibody does not contain the fluorophore, but rather it contains now your enzyme so that a reaction will occur, and that's now your colorimetric detection. Another way is your conjugated antibodies. Sa conjugated antibodies, wala na tayong primary and secondary. Only one antibody that already contains your fluorescence-based detection. Kaya nag-fluoresce or nakikita natin sa microscope. But I guess your conjugated antibodies is more expensive than your fluorescence-based detection. I guess this will be discussed in depth when you reach your IHC na discussion with Dr. Clarion. And I will also listen that kasi never pa akong naka-encounter nito. Diba? It's very amazing. So, that's your basic concepts sa ating IHC. So now let's proceed with the staining of your paraffin section. So we also have a different techniques or strategy on how to stain a paraffin section. So first step na dapat nating malaman is your deparaffinization. So Doc, bakit natin, di ba pag sinabi ng deparaffin, so you are trying to remove the paraffin from your paraffin section. Doc, bakit natin inaalis yung paraffin? Remember that your paraffin wax is poorly permeable to most staining solutions and should therefore be removed from the section prior to staining. So this is usually done by immersing your paraffin section in a solvent. Specifically, ang gagamitin natin is your xylene 2 times at 1 to 2 minutes duration each for sections up to 10 micron thick. So again, deparaffinize using your xylene para matanggal yung ating paraffin since your paraffin is poorly permeable sa ating staining solution. After that, anong gagawin natin, Doc? After that is that we do a sections to alcohol. Ito yung tinatawag natin na sections to alcohol. Ang re-remove naman natin na yun is your xylene. Remember, your xylene is not miscible with aqueous solutions and low-graded alcohol and should therefore be subsequently removed with absolute alcohol followed by descending grades of alcohol. So, i-remove natin ang xylene first by your absolute alcohol and by degrading levels of alcohol. Doc, bakit degrading? Para hindi ma-prevent natin ang damage and detachment of our section. So, again, you deparaffinize using your xylene kasi yung paraffin is not permeable to most of your staining solution. Next, yung xylene na naman ang ating i-remove using your sections to alcohol by your absolute alcohol and then by descending grade of alcohol so that we prevent the damage or detachment of your section. The next step is your alcohol is then replaced with water before your actual staining of section is performed. Such procedure is the exact reverse of impregnation and may be summed up by the phrase of sections to water. So kung meron tayong sections to alcohol, ito naman tinatawag natin na sections to water. Then, the next step is that we will now proceed with your staining. So, i-move ko lang yung self ko kasi hindi nyo makikita. Wait lang. Move ko lang. Okay, dyan. Perfect. So, staining. After staining, the section is again dehydrated with increasing grades of alcohol. So, parang binaliktad lang natin. So, this is now your dehydration. So, increasing grades of your alcohol. Then, the step 5 is that cleared with 2 changes of your silane to prepare the section of mounting since most mountants are miscible in silane. So again, you will now proceed with clearing using your silane. And ayos lang yung silane kasi nga your silane is actually what you call miscible with your most of your mountants. Take note that the second change of your silane here will raise the refractive index of your glass slide, thereby reducing the light refraction during microscopic examination. The stain section may be left in the silane for an indefinite period of time until it is finally mounted sa ating slide. So, ayos lang na malagay natin yung sample natin na stain sa silane kasi it will enhance your refractive index sa ating glass slide. However, the section should not be allowed to stay here in your alcohol for a long time because many stains are usually removed by prolonged immersion in your alcohol. So, ayaw natin siyang ma-stay dito sa alcohol pero ayos lang sa ating silane. So, again, let's review the staining of your paraffin section. First, we have to do deparaffinization because your silane is poorly impermeable sa ating most of your aqueous solution. Then, after that, using your silane, then we will proceed with your sections to alcohol. I-remove natin yung silane because your silane is immiscible by your grading absolute alcohol and then by decrease grading of your alcohol, and then we wash with water, then we stain. After staining, we will now proceed with your dehydration. Lastly, clearing before we mount our sample. So that's the basic strategy when we do your staining of your paraffin section. Okay, move ko na yung self ko. Babalik ko na dito. Ayan. Okay, let's proceed with the methods of staining. For the method of staining, we have the first method, your direct staining. So, direct staining is the process of giving color to the section by using aqueous or alcoholic dye solution. In simple or direct, ang other name for direct is simple. Simple staining, only one dye is used, which is washed away after 30 to 60 seconds prior to drying and examination. Example of this is your methylene blue. So, the molecules that make up basic dyes have a positive charge. This is important because the cell wall and your cytoplasm of the bacterial cell have a negative charge. So, the positive charge dye is attracted to the negative charge cells, enhancing the ability of the stain to stick to and color the cell. So again, basic idea, since your methylene blue is a basic dye ang ating methylene blue, it's a positively charged. And remember that your cytoplasm and your cell wall and your cytoplasm of your bacteria is negatively charged. That's why it will attract. That's why a color, since it's blue, it will present as a blue in color. So ito yung ating tinatawag na simple or direct simple staining. So anong ginagawa? Doc, ano po ito? So you will learn this more when you reach your microbiology. But for explaining you this, ang ginagawa namin, kukuha kami ng colony ng aming bacteria, tapos ilalagay namin dito. Tapos nilalagyan namin ng NSS para ma-distribute yung bacteria. So pinapadry lang namin dito in heat fix. After heat fixing your sample, nilalagyan na namin ng color blue. Ito na yung tinatawag natin na methylene blue. And with the principle that I said a while ago, ito na yung lumalabas. Your bacilli here, stained color blue. Ano ito? These cells, ano ito? These are your WBC, since these are your bacteria. Okay. We also have your indirect staining. Your indirect staining is the process whereby the action of the dye is intensified by adding another agent or a mordant which serve as a link or bridge between the tissue and the dye to make the staining reaction possible. So example of mordants, marami tayong example dito. Kindly take note of this. So in short, your indirect staining, you will need a mordant for the reaction to occur. Meron tayong ginagamit also na accentuator. An accentuator, on the other hand, is not essential to the chemical union of the tissue and the dye. It does not participate in the staining reaction but merely accelerates the reaction. So these are the example also. So ito yung diagram na will help us understand the principle. Ganyan natin. Sorry. So you have your stain. So this is now your tissue and sample. So in indirect staining, you will need a mordant. So your mordant will serve as a bridge to your another agent, which is your stain. So your mordant is integral part of the staining reaction itself, because without this one, no staining possible will occur. So kailangan natin ng mordant for the reaction to occur. On the other hand, your accentuator is not essential. Hindi siya kailangan talagang merong accentuator for the reaction to occur, but rather it will hasten or accelerate the reaction. So ito na yung indirect staining using now your mordant or your accentuator. Okay, balik lang natin. Diyan. Okay, that's your indirect. So may dalawa na tayong na-learn na. Yung direct, just a simple staining using one dye. Yung indirect, you could use your accentuator or your mordant. Let's proceed with your progressive staining. So progressive staining is the process whereby your tissue elements are stained in a definite sequence. So meaning, meron talagang sequence na dapat sinusunod. And the staining solution is applied for a specific period of time or until the desired intensity of coloring of the different tissue element is attained. So that's your progressive staining. On the other hand, your regressive staining, the tissue is first overstained to obliterate the cellular details, and the excess stain is removed or decolorized from unwanted parts of the tissue until the desired intensity of color is obtained. So the most common example here is your routine hematoxylin and eosin staining. So you will encounter a lot of your routine H&E staining. Remember that your regressive is the most common method. Your routine hematoxylin and eosin staining is the most common method utilized for your microanatomical studies of tissues using your regressive staining, which consists of your overstaining the nuclei followed by the removal of your excessive color of tissue constituent by acid differentiation. So, pag regressive staining, think about of your routine hematoxylin staining. Sa regressive, you are thinking about overstaining the sample. So, anong ginagawa namin? May sample, tissue sample or tissue dye. You just overstain your sample. That's your regressive staining. Now, let's talk about your differentiation. So, differentiation or decolorization, it's the selective removal of excess stain from the tissue during regressive staining. So, during regressive, di ba? We do this regressive, we overstain our sample in order for a specific substance may be stained distinctively from the surrounding tissue. Doc, ano po yun? Again, pag sinabi nating differentiation, selective removal of the excess stain from the tissue during regressive staining in order that a specific substance may be stained distinctly from the surrounding tissue. So, example nito, this is the most basic or common example na makikita nyo when you reach your microbiology. So, this is an example of your gram staining. Sa gram staining, meron tayong 4 na stain na ginagamit or 4 na mga reagents na ginagamit. Remember this ha, ang mnemonics na ginagamit natin, ang mnemonics na ginagamit namin is your VIAS. Your VIAS stands for violet or your crystal violet. Secondly, your iodine. Next is your alcohol, and lastly, your safranin. So, VIAS, again, crystal violet, iodine, alcohol, safranin, VIAS. So, this is an example of your gram staining. So, ang gram staining is used for decolorization. So, part ng step ng gram staining is using your decolorization. Ito yung decoloration, the third step. Sige, check natin. First, Doc, ano po ba ang gram staining? So you will learn that in your microbiology. But for at least we have an overview about it, pag sinabi natin gram staining, we will be able to identify whether a certain bacteria is gram-positive or gram-negative. Ano po yung gram-positive? Ang gram-positive is a type of bacteria wherein ang bacteria natin ay nagsistain ng purple. Pag gram-negative is a type of bacteria wherein our cells retain the secondary stain which is your safranin or your counter stain. Doc, bakit po ba nagiging gram-positive or gram-negative? Nagiging gram-positive, it is because of the cell wall of the bacteria. In gram-positive, makapal ang kanyang cell wall because of the presence of your peptidoglycan. And in our gram-negative bacteria, anong nangyayari sa gram-negative bacteria? Meron pa ring cell wall, meron pa ring peptidoglycan, pero manipis lamang ang peptidoglycan. Therefore, it will not able to retain the color of your crystal violet. Ang nare-retain niya lang is yung color ng counter stain or your safranin, which is color red or pink. So again, ano bang step niyan, Doc? Sige, Doc, medyo nalilito pa kami, Doc. So gram staining, ito, first step is your crystal violet. Your crystal violet is your primary stain. Again, your crystal violet, siya yung primary stain natin. Second step is your iodine. Your iodine will serve as your mordant. Di ba, remember, ano nga mordant? Ang function ng mordant is that it will serve as a bridge between your tissue and your primary stain para a reaction will occur. So that's the purpose of iodine. Next, alcohol. Dito na yon gusto kong ma-learn nyo kasi your decolorization is brought about by the alcohol. Alcohol will serve as a decolorizing agent. So ano po bang nangyayari, Doc? Remember, once it is a gram-positive, diba? Remember, gram-positive siya, makapal ang cell wall ng bacteria. Makapal ang cell wall brought about by your peptidoglycan. If it is gram-positive, mareretain niya ang kulay ng ating primary stain which is violet, crystal violet, kasi nga makapal ang peptidoglycan. It will have a great affinity with our peptidoglycan. So wala tayong problema dyan. In short, when you decolorize it, you cannot remove the primary stain. Again, you cannot remove the primary stain. However, if it is gram-negative, anong nangyayari? Manipis lang yung cell wall ng ating gram-negative, kaya maliit lang yung peptidoglycan, kaya nade-decolorize siya. It cannot retain the primary stain, which is your crystal violet. So, nawawash out yung violet na color. So, as what you can see here, nawawash out yung violet na kulay. Now, after that, you will proceed with your next counter stain or your stain, which is your safranin. Sa safranin, so you will add your safranin na reagent and it will colorize now your bacteria na natanggalan ng kulay. So because it will serve as your counter stain, that is why your gram-negative will be colorized with your safranin as color pink or red. That's why sa microscope, nakikita natin na color pink yung ating gram-negative. And your gram-positive, Dok, bakit hindi po nakukulay ang color pink since naglalagay naman tayo ng color pink or safranin sa atin? Remember, meron na siyang crystal violet dito, and your crystal violet has already a strong affinity with your peptidoglycan or your cell wall. Plus, nag-add ka pa ng mordant which makes the reaction stronger. That is why your gram-negative will only be the one colorized by your safranin, kaya nagiging gram-negative siya. So I hope that you understand the basic concept. So your decolorization, you just want to remove the excess stain in your regressive staining in order for that specific substance may be stained distinctly from the surrounding tissue so that you will be able to identify this gram-negative. That's the basic concept in your decolorization para ma-identify mo yung specific na bacteria. Again, di ba, if i-compare natin to sa ating simple staining, ano nga sa simple staining? Methods for simple staining, impart same color to all bacteria. Kung simple staining lang, di ba, yung ginawa natin na methylene blue, lahat color blue. Like this one, lahat purple. Pero on the other hand, in differential staining method, it imparts a distinctive color only to a certain types of bacteria. For example, your gram-negative. So, don't you worry, you will learn more pagdating nyo sa microbiology. So, your differential staining uses more than one chemical stain to better differentiate between various microorganisms or structure or cellular components of a single organism. Again, this is the concept that I discussed to you a while ago. So, it is a differential. Bakit differential, Doc? Because we will be able to identify various microorganisms or structures sa ating tissue sample. For this part, we will be able to identify whether gram-positive or gram-negative. So, this is an example of your gram stain procedure. So, tingnan lang natin yung diagram. Or at least you have an idea about this one. So, unang-una, kuha ka ng glass slide. Tapos, kuha ka ng colony mo with NSS. Tapos, spread out mo lang sa ating glass slide. Smear the bacteria with the slide. And then, you do heat fixing. So, ang purpose ng heat fix para mas lalong dumikit yung ating specimen sa ating glass slides. So, you will learn this more in your microbiology. Tapos, our primary stain, which is your crystal violet. So, si crystal violet is your primary stain. It will stain those with gram-positive na mga cell wall. Yung mga makakapal ang peptidoglycan. Then, you will wash that with water. Then, after that, you will add now your mordant. Remember, your iodine will serve as a mordant that will serve as a link between your tissue and your sample para reaction will occur. Then, you will do na, ito yung gusto ko ma-learn nyo, yung decolorize with your alcohol. So, tatanggalin na yung stain na hindi kumakit. And then, you do decolorization. Ganyan lang, through side lang. Tapos, you wash that with water. And lastly, you will add now your counter stain, which is your safranin. So, your safranin now will stain your gram-negative. So, nagiging color pink ang gram-negative. Kasi technically, your safranin is color pink. Then, you wash it again and then let it dry. So, tingnan lang natin sa microscope kung anong pinapakita ba nila. I'm not sure. So, you blot dry. Actually, ang ginagawa lang namin, pinapa-air dry lang namin. So, pinapaslant lang namin yung slide para air dry lang. Kasi napakatakot pag naka-blunt dry ka, patanggal yung specimen. Baka didikit siya. So, ito yung nakikita namin, violet because of the gram-positive bacteria, stain purple because of the thick peptidoglycan layer which retains the crystal violet iodine complex. So, ito yung sinasabi kong gram-positive. So, yung gram-negative bacteria appears pink because they have a thin peptidoglycan layer which does not retain your crystal violet but it is stained by your safranin. So this is the basic concept of your gram staining. So let's proceed with your metachromatic staining. So for metachromatic staining, it is a staining technique that entails the use of your specific dyes which differentiate your particular substances by staining them with a color that is different from that of the stain itself. So ito yung tinatawag natin na metachromatia. So these are the examples of your mga metachromatic stains. Next, you also have your metallic impregnation. So for metallic impregnation, it is a process where your specific tissue elements are demonstrated not by stains but by colorless solutions of metallic salts which are thereby reduced by the tissue, producing an opaque, usually black deposits on the surface of the tissue or bacteria. So, example nito, meron tayong gold chloride and your silver nitrate. So, for this diagram, tingnan natin. So, you are using now your silver nitrate or your chemical, your silver. For example, your silver nitrate stain is being reduced by your argentaphine cells. So remember your argentaphine cells, which is found in your melanin and your intestinal glands. So when that argentaphine cells reduce the stain, your silver stain, it will now form your black deposits under the microscope. So always remember that your metallic impregnating agent is different from a stain because it does not absorb by the tissue. It is a precipitate or as a reduction product in a certain tissue components. Again, yung metallic impregnation natin is different from a usual stain because it does not absorb by the tissue. Hindi siya ina-absorb ng tissue, but rather it is a precipitate or as a reduction product ng certain tissue components. Kaya nagkakaroon tayo ng black deposit sa ating microscope na nakita. The next technique is that we have your vital staining. So your vital staining, we have two different types of vital staining. Meron tayong tinatawag na intravital staining and your supravital staining. First, let's define vital. So vital is the selective staining of living cell constituents. So we are talking about your living cell, demonstrating cytoplasmic structures by phagocytosis of the dye particle, which is your cytoplasmic phagocytosis, or by staining of pre-existing cellular components, which is your true vital staining. So, pag sinabi natin vital staining, which this refers to a living cell. So, meron tayong dalawang example. We have your intravital or supravital. Pag sinabi natin intravital staining of living cells is done by injecting the dye into any part of the animal body, either intravenous, intraperitoneal, or subcutaneous. So, you are introducing now the stain towards the body, inside the body. You are injecting it inside the body. So example of these are your lithium carmine and your India ink. For your supravital staining naman, it's a different method because it's a method of staining used in microscopy to examine living cells that have been removed from an organism. So you take out the living cell outside the body. So example of this is your butyral red, Janus green, Trypan blue, Nile blue, thionin, and your toluidine blue. Just remember that nucleus not demonstrated in your vital staining. The nucleus is not demonstrated due to the resistance to vital stain. So in short, pag nucleus, hindi natin nakikita sa vital stain because your nucleus will resist the vital stain. Pero pag Doc, gumagamit po ako ng vital stain but I was able to see the nucleus, then that would only mean that the nucleus already dead. So, meron din tayong sinasabi na neutral red na stain. Yung neutral red is probably the best vital dye. So, remember that. And also, your Janus green especially is recommended for mitochondria. So, check natin ang example ng supravital stain. So, we have your this is an example of your supravital stain for your reticulocyte staining. So, you will also learn this in your hematology. So, for reticulocyte staining, ang ginagamit natin is your new methylene blue or your brilliant crescel blue. Please remember this because this is a very common na mga staining na ginagamit natin not only in histopathology but also in other subjects such as your hematology. So, Doc, ano po bang ginagamit? Bakit po tayo? Ano po bang reticulocyte count? Ano po ba yung reticulocyte staining? You will learn that in your hematology that your reticulocytes are your immature red blood cells that is produced by your bone marrow. And these reticulocyte cells are being released in your peripheral blood where they mature into red blood cells within 1 to 2 days. So, bakit tayo gumagawa ng reticulocyte counting, Doc? Bakit natin kinakount yung reticulocyte count? So, we do counting of your reticulocyte count because any increase or decrease in your reticulocyte count can be an indicator of your erythropoiesis activity. Pag sinabi natin erythropoiesis, it's the formation of your blood cells or red blood cells. So, we want to check whether your bone marrow is able to produce or not produce your red blood cells, especially relative to anemias and bone marrow dysfunction. So, Doc, remember remember your supravital staining, you are using your live red blood cells because ang ginagawa lang namin is that kukuha ka lang ng blood sample tapos ilalagay mo lang sa glass slide and then you just put your new methylene blue or brilliant crescel blue tapos lalagyan mo ng cover slip tapos titingnan mo lang under the microscope. And this is now your reticulocyte count because of the presence of your reticulin fibers. Reticulin fibers is commonly seen in your reticulocyte count. So, yan yung magdi-differentiate from your mature red blood cells without your reticulin fibers and with your reticulocyte counts with your reticulocytes with your reticulin fiber. So these are your reticulocytes. So we have also your hematoxylin and eosin staining. So this is a very specific types of staining na tayo. We have your H&E staining. So your H&E staining is the cornerstone of tissue-based diagnosis. It stains your thin tissue sections to visualize your tissue morphology. Hematoxylin dye, remember that your hematoxylin dye stains your cell nuclei blue because it binds to the dye strongs to acid, and your eosin dye to stain other structures pink or red. Diba yan yung sinabi ko kanina, yung principle na gusto kong malaman nyo. Your routine H&E staining in paraffin embedded section is used as your regressive staining. Just remember that most fixatives can be used except your osmic acid solutions, which will inhibit your hematoxylin. Once you are using your routine H&E, dapat tandaan nyo yung fixative na ginamit nyo. Pero most of the fixative naman are pwede except lang your osmic acid solution. So remember this one. And also, in your H&E staining of your frozen section for rapid diagnosis, we are using now your progressive staining. However, sa H&E frozen section, it is less favored regressive staining due to mas less favored yung progressive staining kesa sa regressive staining because of the it will have a difficulty of producing sufficiently intense progressive staining of your cell structures without staining other parts, thereby resulting in diffused color and obscured detail. So mas mahihirapan tayo if we are using now your progressive staining sa frozen section compared to a regressive staining. Kasi sa regressive staining, mas easier natin na nakukuha yung color na gusto natin. I guess we are also down to our last slide. We also have your restaining of your old section. So restaining of your old section, basic lang. You also have to, the old slide is usually immersed in your xylene for 24 hours or gently heated until mounting medium bubble. So then your cover slip is lifted. So tatanggalin mo yung cover slip and placed in your xylene for 3 minutes. Then brought down to water. Then placed in your 0.5% potassium permanganate solution for 5 to 10 minutes, then rinse again in tap water, Then immerse in 5% oxalic acid for 5 minutes or until completely decolorized. Then you wash that with running tap water for another 5 minutes. Then now you can stain your old section. Doc, bakit ba natin kailangan i-restain your old section? So may mga times na you have to compare the previous section of a certain patient. So kumuha ka ng tissue sample dati, and then for following years, you check it and compare with the previous section for your proper diagnosis. So that ends my video lecture. It's only a very short discussion. And if you have any question, please do not hesitate to ask or PM me. So for your sync class also, it will be after na lang ng inyong quiz so that we can answer directly the quiz. So that ends. Thank you everyone. I hope you had also fun learning a new topic. Thank you everyone.